So let's take a look at how mitosis happens in a eukaryotic cell. Now remember that the main phase that cells can be found in is interphase. And cells spend a lot of time in interphase because they're just doing their normal cell thing. But let's take a look at what happens if a cell is going to move on past interphase into those other phases of cell division. Now remember, during interphase, first there's that phase of G1 where the cell's doing its normal thing and growing, and then after G1 comes S phase. Now here is our DNA in the nucleus. So this is our nucleus of a cell in interphase, and the DNA, remember the DNA is in this sort of loose, long, open, squiggly form. We're going to call that chromatin. All right, chromatin. So DNA is in chromatin form. During the S phase of interphase, all of this DNA is going to get copied or replicated. So we're going to see the amount of genetic material in there double. Let's say this is a cell that has 6 pieces of DNA. Now it still has 6 pieces of DNA, but it has 6 replicated pieces of DNA. After the S phase, the cell will move into G2 phase. During G2 phase, it maybe grows a little bit more, gets a little bit better prepared for cell division. These are all phases of interphase. Now from an outside viewer, you're not going to see too much happening. So on your paper, go ahead and sketch this cell for a cell in interphase. It's got DNA in the nucleus in the form of chromatin. We know that DNA replication happened, but we're not actually going to see chromosomes yet because that's just not the form the DNA is in during interphase. All right, so now we are ready to divide the cell. The cell thinks that what the material in the nucleus is so important that it's going to actually do this form. It's going to divide up the nucleus separate from the rest of the cell, and it's going to take very, very special care with the nucleus. So the first phase of dividing up the nucleus is called prophase. Prophase. And I like to think of prophase as being like preparing to divide the nucleus. So prophase, prepare, PP. And the first thing that's going to happen is the cell wants to sort of really just organize this DNA so it can make sure it divides it up evenly. And so what it's going to do is a couple of things. It's going to actually break down the nuclear membrane and spread the DNA all around the cell. Now, as the DNA gets spread around the cell is actually going to take that DNA and condense it. Remember, we've been talking about how when this long strand of DNA gets condensed, it really gets wrapped around those histone proteins until it sort of looks like nice little fat sausages instead of long spaghetti. So I'm going to sort of shorten this process up and show you a cell with 6 pieces of replicated DNA, all spread around the nucleus in a different form. This different form, so now the DNA isn't chromatin, now DNA is in, chromosome form, right? And specifically, these are replicated chromosomes. And you'll notice that because they each have, there are 2 identical chromosomes. Remember, a chromosome really should just look like kind of one single thing. But there are 2 identical copies of chromosomes held together in the middle. So let's remember our labels here. If this whole thing is a replicated chromosome, remember that that middle piece there that holds it together in the middle is a centromere, and that each half of a replicated chromosome is called a chromatid. So during prophase, that nucleus goes away and the chromatin turns into these replicated chromosomes. A few other things happen. We get these areas of the cells that start organizing called the poles. And at the poles, sometimes you get 2 structures called centrioles, depends on what type of cell it is. We're not going to worry too much about that word, but we get these poles. And also from the poles, we get special sort of stringy fibers called spindle fibers. And the spindle fibers attach to each chromosome at the centromere. And why do they do that? Well, these spindle fibers here, when they attach to the chromosomes, they're going to be able to kind of move them around. So if you imagine some strings, they can be able to pull these chromosomes around wherever they need to in the cell during the next few phases. So this is prophase. Go ahead and sketch prophase on your note sheet. There are 6 replicated chromosomes in the cell during this phase here. So let's move on to the next phase, which is metaphase. And I like to remember metaphase. It starts with an M, and that what happens in metaphase is that all the chromosomes get moved to the middle. So those spindle fibers, remember those spindle fibers, how they were attached to our replicated chromosomes? They're going to take and kind of push and pull these chromosomes until they're all lined up down the middle of a cell, like soldiers. Now, I know it's hard to see, but we've got all those chromosomes lined up down the middle. I have to imagine my little imaginary thing here a little bit better. But you should see all 6 chromosomes lined up in a line right down the middle of a cell. Those spindle fibers are attached, remember, still to the centromeres of all these chromosomes. Go ahead and draw what happens in metaphase. Draw 6 of those replicated chromosomes lined up right down the middle of the cell. That's metaphase. Notice that if these are the poles, the chromosomes line up in what would sort of be like the equator. Like if this is the North Pole, this is the South Pole, this would be the equator. All right, they don't go from pole to pole. They're kind of stretched out in the middle between the 2 poles. All right, so now we're ready for the third phase of dividing up the nucleus, which is called anaphase. There we go. Anaphase. And in anaphase, each of these replicated chromosomes is going to get pulled apart. So the spindle fibers are going to pull so hard on each of these 2 halves of the replicated chromosome that each replicated chromosome is going to get pulled apart and start getting pulled towards the poles, or the opposite sides of each cell. And that's really all that happens during anaphase. During anaphase, each replicated chromosome gets ripped in half and starts moving towards the opposite ends of the poles. Anaphase apart, AA. So the final phase of dividing up the nucleus, oh, and if you didn't, go ahead and draw that on your paper as well. You should see 6 single chromosomes going one way and 6 single chromosomes going the other way. We no longer have replicated chromosomes. We have those single chromosomes now. And telophase, I like to think telophase, the end of dividing up the nucleus. What's going to happen is these chromosomes continue to get pulled by the spindle fibers towards the opposite poles. And what's going to happen is our spindle fibers are then going to get broken down by the cell. The cell doesn't need them anymore once it's finished pulling these to opposite sides. And then it's going to take and put a nucleus around each set of single chromosomes. So you'll notice we started off with 6 replicated chromosomes, and now what we have is 2 nuclei. Each one has 6 chromosomes in it, but there's 6 single or normal sort of regular chromosomes in there. This is telophase. Now towards the end of telophase, each of these chromosomes is going to sort of unwind and loosen back up and turn into chromatin. But before I do that, I want you to go ahead and sketch this as being telophase, showing the 6 single chromosomes, each inside its own nucleus. Now, kind of at the same time that telophase is happening, there's another phase that's also going to be happening that has nothing to do with dividing up the nucleus, and it has everything to do with dividing up the rest of the cell. And that is cytokinesis. And cyto in cytokinesis refers to dividing up the cytoplasm. So during cytokinesis, we're going to divide up the cytoplasm. Now, before I do that, we're going to show that each of those chromosomes got uncoiled. I had to separate my piles here. So there's our chromatin back in the nucleus. In cytokinesis, the cytoplasm is going to need to be split up. Now in plant cells will just sort of build a wall right down the middle. If this is an animal cell, what will really happen is that the cell membrane will kind of start to pinch in, and it'll keep pinching in until there's a new piece of cell membrane right in between the middle here, so that we end up with 2 completely different cells. So here's one cell membrane, here's the other cell membrane, and we have 2 separate cells, each with an exact full copy of DNA. Our starting cell had 6 chromosomes. Now we have 2 different cells, and they each also have 6 chromosomes. Go ahead and sketch this on your sheet. That is mitosis.